Okay, let's unpack this. Think about pain for a second.
Mm.
Not just, the temporary sting, but that deep, persistent ache.
Hmm.
The kind that sticks around long after an injury has supposedly healed, or maybe the type that just seems to creep in and get worse as the years go by.
Yeah,
It can really reshape your life, can it?
it absolutely can. Chronic pain, especially that nerve-related pain, it's a huge challenge.
Right.
And the fact that it becomes more common as we age or after nerve damage, well, it suggests there's some kind of fundamental process linking those things together
Yeah.
at the cellular level.
Yeah, like something going on underneath it all.
Exactly. But finding exactly what that cellular process is, well, that's been tricky.
Right. What if there's a surprising cellular culprit kind of hiding in our nerves, contributing to this specific problem?
Well, that's where this new research comes in.
That's the fascinating journey this deep dive takes us on. We're cracking open a brand new piece of cutting-edge research published in the journal Nature Neuroscience.
Yeah, the paper is titled Aging and Injury Drive Neuronal Senescence in the Dorsal Root Ganglia.
Ah,
It looks deep into the cellular changes happening in a really critical part of your peripheral nervous system.
okay.
These clusters of nerve cell bodies called the dorsal root ganglia, or DRG.
DRG, got it.
These are essentially relay stations. They receive sensory information, including pain signals, from your body, and then send them on towards your spinal cord and brain.
Okay, so our mission today is to take this complex scientific study and, well, translate its most important findings just for you.
Mm.
We want to understand what they discovered about neuronal senescence. That seems like a key term here.
It really is.
What is it? How does aging and nerve injury cause it, specifically in these pain-signaling neurons in the DRG?
Right.
And the biggest question, I guess, could these senescent neurons actually be causing pain? And if so, could we potentially target them, find new ways to treat it?
That's the ultimate goal.
It feels like understanding a potential fundamental link between getting older, getting injured, and experiencing these chronic sensory problems.
Exactly. So let's start with that core concept.
Yeah.
Cellular senescence. You might have heard of it in the context of aging, maybe?
Vaguely, yeah. Cells getting old.
Sort of. Simply put, it's a state some cells enter where they stop dividing. That's a classic hallmark. But crucially, they also become resistant to dying off.
To dying off.
Yeah, they resist the normal programmed cell death process called apoptosis.
So they just hang around indefinitely.
Precisely. And it's not just passive hanging around either.
Right.
While they're in this state, they become metabolically active and start releasing this cocktail of molecules.
Okay.
This is known as the senescence-associated secretory phenotype, or SASP.
SASP.
And many of these SASP factors are pro-inflammatory, or they can affect neighboring cells. This whole process, senescence, happens throughout the body as we age, or after various stresses, including injury.
But hold on, we're talking about neurons here. My understanding is that mature neurons generally don't divide once they're, developed.
That's right.
So how can they undergo senescence if that's usually defined by stopping division?
That's a really insightful point. And yeah, it was a challenge for researchers for a long time. Neurons are postmitotic, meaning they don't divide.
Right.
However, this research and other recent work introduces the concept of postmitotic cellular senescence, or POMICUS.
Pumice. Okay.
So while they don't divide, these neurons do acquire other key features of senescence.
Ah, like which features them?
Well, they start expressing classic senescence marker proteins, specifically ones called P21 and P16.
P21 and P16, like little flags.
Exactly. Think of them like flags that scientists can look for inside a cell. Say, okay, this cell is in a senescent state.
Gotcha.
And just like other senescent cells, these neurons develop that SASP we mentioned. They start releasing factors. The paper focuses heavily on one in particular,
IL-6.
IL-6.
Yeah.
It's a potent pro-inflammatory molecule. Now, the researchers discussed the possibility that maybe, temporarily entering this state helps a damaged neuron avoid trying to divide, which could be disastrous, and instead just stay alive after stress.
So maybe a protective thing initially?
Possibly. But their findings strongly suggest that if this state persists, if it sticks around, it becomes dysfunctional and actively contributes to problems, like pain.
Okay, so it's possible for even non-dividing neurons to become senescent, marked by these proteins and releasing inflammatory stuff like IL-6. What did the researchers find about whether this actually happens in the DRG as we age.
Right, that was one of their first big questions. They looked at the DRG from aged mice, essentially elderly mice,
Yeah.
around 20, 24 months old,
Okay.
and they compared them to young adult mice, about 11, 16 weeks old.
And was there a difference?
Oh yeah, a very clear one. They found a definite accumulation of senescent neurons in the aged mice's DRG.
How could they tell?
Well, one method they used was staining for something called SAUGAL activity. It's a common test where senescent cells literally take on a blue stain under the microscope.
Ah, so you could see them.
Exactly. And they saw many more blue stain cells in the aged DRG.
And those protein flags, P16 and P21, did they check for those too?
Yes, absolutely. They confirmed that the percentage of neurons expressing those key senescence markers, P16 and P21, was significantly higher in the aged mice compared to the young ones.
So aging alone seems to bring on this senescent state in DRG neurons. What about that pro-inflammatory SAP you mentioned, the IL-6?
Right, that's where the IL-6 comes in. The aged DRG didn't just have more neurons expressing IL-6 in general,
Okay.
but particularly they saw a significant increase in neurons that were both expressing P21, the senescence marker, and IL-6, the inflammatory factor.
Ah, so the senescent cells are the ones making the IL-6.
A larger fraction of them, yes. In aged mice, more of the IL-6 producing neurons were also positive for either P21 or P16. It strongly suggests that these aging senescent neurons are a significant source of this pro-inflammatory molecule right there in the DRG.
Wow.
They even found increased levels of IL-6 protein circulating in the blood plasma of aged mice.
Really? So it might even have effects beyond the nerves.
It suggests this local inflammation in the DRG might have systemic effects, yeah.
That's a powerful link, aging causing senescent neurons and those senescent neurons pumping out inflammation. Now, given that nerve injuries often lead to chronic pain,
Right.
what did they find about injury's role in all this?
Good question. To study nerve injury, they used a well-established experimental model in mice called spared nerve injury, or SNI.
SNI.
Yeah, this procedure involves damaging specific nerves in a way that mimics the complex nerve damage seen in humans, which often results in chronic neuropathic pain.
Okay, and when they did this SNI procedure in young mice, what happened to their DRG neurons? Did they become senescent too?
Yes, the nerve injury strongly triggered neuronal senescence in the DRG, even in these young mice.
Even in young ones.
Yep. They use techniques like qPCR, which measures how much specific genes are being expressed, basically how active certain genes are.
Huh.
And they saw a significant increase in the gene activity for P21 and P16, those senescence markers.
Okay.
Plus, increased activity for several SASP factors, including IL-6, IL-1, and CCL2, about 3 weeks after the injury.
3 weeks later.
And importantly, they confirmed that it was primarily the neurons themselves showing this increased gene activity, not other cell types in the DRG.
Got it. Did the senescence markers appear right away after injury, or was there a timeline? Like, how quickly did this happen?
Yeah, there was a distinct time course. P21 expression seemed to spike relatively early, around 7 days post-injury, and then it stayed high.
Okay, P21 early.
But P16, on the other hand, showed a more gradual increase over time after the injury.
Interesting. So different timings. And how did being older affect this injury response? Did age change things?
Yes, and this is a key interaction they uncovered. Aged DRG neurons, which, remember, already started with higher baseline senescence levels.
Right, from the aging process itself.
Exactly. They seem to transition into that P16 positive senescent state more quickly after injury compared to young neurons.
So aging kind of primes them.
It looks like it. Aging doesn't just cause senescence on its own. It also seems to make the neurons more susceptible to injury-induced senescence, especially that P16 positive type. And some studies link P16 to a more stable or late-stage senescent state.
Okay, so both aging and injury drive DRG neurons to become senescent, but are these senescent neurons only the ones whose axons were directly damaged in the injury? Or does it affect other neurons in the area too?
That's a crucial question the researchers investigated, and the answer is really interesting. They used a marker protein called ATF3.
ATF3?
Yeah, ATF3 is expressed by neurons whose axons have been directly injured. It's like a flag for I'm hurt. As expected, they found that the neurons positive for ATF3, the directly injured ones, did show increased expression of P21 and P16 after the injury.
Makes sense.
In fact, a large majority of these injured ATF3 positive neurons co-expressed one or both of the senescence markers.
So the primary injured neurons are definitely going senescent. No surprise there, maybe.
Right. But here's the critical finding. They also found that non-injured neurons, those that were negative for that ATF3 marker in the injured DRG, also started expressing P21 and P16.
Wait, the ones that weren't directly injured?
Exactly. The bystanders, so to speak.
Non-injured neurons in the area becoming senescent. How could that happen?
Well, this points strongly to the idea of bystander senescence.
Bystander senescence,
Remember when we
okay.
talked about the SAP factors released by senescent cells?
Yeah, the inflammatory stuff like IL-6.
Right. The discussion section of the paper highlights how these factors like IL-6, released by the primary senescent cells, probably the injured neurons themselves, can act on their neighboring cells, including other non-injured neurons. It's like those primary senescent cells send out molecular signals that basically convince their healthy neighbors to also enter that dysfunctional senescent state.
Even though they weren't directly damaged. Wow.
Yeah, it's a kind of domino effect, creating a larger pool of senescent cells in the injured area than just the initially hurt ones.
That bystander effect sounds really significant. So, okay, we have aging and injury driving senescence in both directly injured and these bystander neurons in the DRG. How does this growing pool of senescent neurons connect specifically to pain? What type of neurons are these senescent cells predominantly?
Good question. They looked at the physical characteristics. They found that neurons expressing senescence markers, particularly those expressing IL-6, were generally small diameter neurons.
Small diameter neurons. And those are precisely the category that includes nociceptors, right? Those specialized nerve cells that detect potentially harmful, painful stimuli.
Exactly, nociceptors. So they then specifically investigated TRPV1 positive neurons.
TRPV1, that rings a bell.
Yeah.
Heat and chili peppers.
That's the one. TRTV1 is a well-known marker found on a significant population of small diameter nociceptors. These are the neurons activated by heat, acidity, inflammatory molecules. They contribute heavily to pain sensation.
Okay.
And what they found was that these TRPV1 positive neurons readily co-express the senescence markers P21 and P16 and also IL-6.
So the pain sensors themselves are becoming senescent.
Yes. And importantly, the co-expression of these markers in TRPV1 positive neurons increased significantly after nerve injury in both young and aged mice.
Okay, that establishes a direct link then. The senescent population includes, and seems to expand within, the very neurons responsible for sensing pain.
Precisely.
So we have pain-sensing neurons becoming senescent, and they're releasing inflammatory signals like IL-6. How does that biochemical soup, I guess, actually translate into the physical experience of heightened pain sensitivity or chronic pain? What's the mechanism?
Right. How does it make things hurt more? This is where their electrophysiology experiments are incredibly telling.
Electrophysiology measuring electrical activity.
Exactly. They measured the electrical activity of DRG neurons, looking at how they fire action potentials, those electrical signals neurons use to communicate.
And
They found that neurons positive for senescence markers like P16 and IL-6, and even some P21 positive neurons, were often located in clusters that included high-firing neurons,
high firing, meaning
meaning neurons that generate a rapid burst of electrical signals over 100 action potentials in response to stimulation. Basically, they are highly excitable, easily triggered.
so the neurons hanging out near the senescent ones, or maybe the ones that are senescent, are prone to being overstimulated. That definitely sounds like a mechanism for pain, like a nerve on edge.
It certainly does. They also show that neurons expressing these senescence markers shared physiological characteristics common to nociceptors, like having wide action potentials compared to other types of neurons.
Okay.
But the most direct evidence for the mechanism came from applying IL-6 directly to cultured, aged DRG neurons in a dish.
So they took cells out and just added the IL-6.
Yep, to see what it does on its own.
And what effect did that have?
When they exposed these aged DRG neurons to IL-6, they found that it significantly increased the firing, the electrical excitability of these neurons.
Okay, so IL-6 makes them fire more easily.
Yes. But what's particularly critical here is that many of the neurons that became more excitable after seeing IL-6 did not themselves express IL-6.
Oh. Okay, I see. So the IL-6 being released by a senescent neuron isn't necessarily just affecting itself.
Right.
It's directly making its neighbors more electrically excitable.
Exactly. This is strong experimental support for that paracrine action we talked about, the SAP factors from senescent neurons acting on surrounding healthy or perhaps less senescent neurons.
It's that bystander idea again.
Yeah. This IL-6-driven hyperexcitability in neighboring neurons is a highly probable mechanism by which senescent cells contribute to the heightened pain sensitivity and chronic pain seen in the DRG, particularly after injury or with age.
Wow. This is a remarkable picture they're painting. Identifying these specific, dysfunctional, hyperexcitable, pro-inflammatory senescent neurons accumulating in the DRG. If these cells are part of the problem, it naturally leads to the question, can we get rid of them?
Right.
This sounds like the perfect scenario to test senolytics.
That's precisely the next step the researchers took. Senolytics are a class of compounds specifically designed to selectively target and eliminate senescent cells.
How do they do that?
They basically trigger their programmed cell death, apoptosis, but primarily in the senescent cells, leaving most healthy cells alone.
Okay. Which senolytic did they use in this study?
They use a compound called ABT-263, also known sometimes as navitoclax. It's a well-studied senolytic.
ABT-263. And when did they give it to the mice?
They administered it to mice starting 3 weeks after the nerve injury. That was a time point when they had already established that senescent neurons had clearly accumulated in the DRG.
Makes sense. Let the senescence build up first. And did the treatment actually clear out the senescent cells?
Yes, it did. Treatment with ABT-263 led to a significant increase in cells undergoing apoptosis in the DRG. They used a marker called CC3+ for that.
Okay, so more cell death.
But crucially, a higher percentage of these dying cells were those that were co-expressing the senescence markers P16 and P21.
Ah, so it was targeting the right ones.
Exactly. This provided clear evidence that the drug was selectively targeting and eliminating the senescent cell population in the DRG.
Okay, so it worked at the cellular level, removing senescent neurons. But the big question, did removing these cells actually translate into helping the mice with the pain-like behaviors caused by the nerve injury? Did they feel better?
This is really the most exciting part, I think, from a therapeutic perspective. The ABT-263 treatment significantly improved pain behaviors in the nerve-injured mice.
Really? How did they measure that?
Well, they saw a clear reduction in mechanical allodynia. That's the condition where normally non-painful stimuli, like a light touch with a filament, become painful. The treated mice reacted less.
Okay, less sensitivity to touch.
Right. And the mice also showed improved weight bearing on the injured limb.
Meaning they were putting more weight on it.
Yes, indicating they were experiencing less pain and were more comfortable using that leg.
That's a huge finding. Getting rid of senescent cells reduced pain behaviors? Was there any difference in how well it worked between the young mice and the aged mice?
Yes, and this is a critical takeaway, especially thinking about humans. While the senolytic treatment helped both groups, the improvement in pain-like behaviors was notably more effective and sustained in the aged mice compared to the young mice.
More effective in the older ones?
Yeah. For instance, that reduction in mechanical allodynia, it was more pronounced and lasted longer in the older animals.
Wow.
This strongly suggests that targeting neuronal senescence might be a particularly promising strategy for chronic pain that is associated with or worsened by aging.
That makes sense if older individuals have more baseline senescence to begin with.
Exactly. And they also confirmed, importantly, that the senolytic treatment didn't negatively impact normal sensory or motor function in the uninjured limb,
So it seemed pretty specific to the problem area.
suggesting some specificity for the dysfunctional state, yes.
So the story from the mouse experiments is compelling. Aging and injury cause senescent neurons. They're linked to pain pathways. They make things hyperexcitable. And removing them improves pain, especially in older mice. But how confident can we be that this applies to humans? It's mice, after all.
Right, the million-dollar question. The researchers took important steps to see if these findings hold up in human tissue. Getting samples from people with confirmed nerve injury is understandably challenging,
Sure.
but they were able to look at post-mortem human DRG tissue.
From donors after death.
Yes, to see if the accumulation of senescence markers with age, which they saw so clearly in mice, is also happening in humans.
And what did their human data show?
They saw clear evidence mirroring the mouse results. There was increased expression of P21 and P16 in human DRG neurons from older donors compared to younger ones.
Okay, same markers increasing with age.
Exactly. They also found more IL-6 positive neurons in aged human DRG, and again, a striking increase in IL-6 positive neurons that also co-express P21 and or P16.
So the senescent inflammatory profile matches.
It does, with a particularly notable increase in neurons positive for all three markers, P21, P16, and IL-6. In the aged human DRG.
Okay, so aging causes similar senescent changes in human DRG neurons as in mice. What about the injury link in humans? Did they find anything there?
Well, direct injury samples are scarce, as I said,
Mm-hmm.
but they looked at ATF3 positive neurons, remember the marker for injured neurons?
Right.
They looked for those in these post-mortem human samples, and they found that injured human neurons did co-express P21. And interestingly, this co-expression was even more prevalent in the aged human samples compared to the younger ones that happen to have some injured neurons.
So again, age seems to make human neurons more prone to becoming senescent after injury, just like in the mice.
It aligns very well with the mouse finding, yes.
I think you mentioned they also looked at some existing human data sets, like gene expression data.
Yes, that's right. They leveraged publicly available human DRG RNA sequencing data. RNA-seq gives you a snapshot of all the genes that are active within cells at a given time.
Okay.
Reanalyzing one dataset confirmed an age-related increase in the activity of the genes for P16 and P21 in human DRG neurons.
Consistent again.
And a reanalysis of another dataset, this one from patients with or without chronic pain following spinal cord injury, found something quite compelling.
What was that?
They found a significant association between a specific senescence gene signature, basically, a known pattern of gene activity found in senescent cells, and the DRG samples from patients who reported pain.
So the genetic fingerprint of senescence was linked to having pain in humans.
That's what the association suggests, yeah.
And similar to the mice, were these senescent human neurons the pain-sensing type? Small diameter, TRPV1?
Yes, the human data again supported the mouse findings on that front, too. Senescent human DRG neurons were generally small diameter neurons. And importantly, TRPV1 positive human neurons, those key pain-sensing nociceptors, showed higher co-expression of P16 and P21 in aged samples compared to younger ones.
Wow. So taken together, the picture is remarkably consistent across both the mouse model and the human tissue analysis.
It really is quite aligned.
Aging and nerve injury drive sensory neurons in the DRG into this senescent state. These senescent neurons include the critical pain-sensing types like TRPV1 neurons. They appear to contribute to pain, likely by releasing inflammatory factors like IL-6 that make neighboring neurons overactive, more excitable.
That bystander effect seems key.
And maybe most excitingly, removing these senescent cells with a senolytic drug showed significant promise in reducing pain behaviors, particularly in older animals,
Yeah,
which aligns perfectly with the human evidence that senescence accumulates in the DRG with age and seems linked to injury and pain states.
absolutely. This research offers a really powerful new way of thinking about chronic pain, especially the kind that often accompanies the aging process or just persists long after an initial injury.
Yeah.
It suggests that maybe it's not just a static consequence of damage or getting older, but involves these specific, dynamic, dysfunctional cells that might actually be targetable.
It's not just wear and tear. It's an active cellular process gone wrong.
Potentially, yes.
This deep dive into the science certainly opens up a lot of avenues for future thought. Thinking about this newfound link between aging, injury, these specific senescent neurons, and chronic pain, well, it raises a compelling question for you to consider. If clearing out these dysfunctional senescent cells can alleviate chronic pain, particularly in older individuals, could this cellular strategy be applied more broadly? I mean, given that cellular senescence is increasingly implicated in many age-related diseases, could targeting these specific cells in different tissues be a general approach? Not just for pain relief, but potentially for promoting healthier aging overall.
That's the big picture question, isn't it?
It really makes you rethink what getting older means at a cellular level, doesn't it?